Hello, my name is Paola Binda and I would like to present some work we've done in the lab on non-trial based experiments, which basically means pupillometry experiments where there is no underlying trial structure or the trial structure is ignored for the purpose of the analysis. Now, since the focus here is on the method, I thought perhaps it's good to start by motivating our work. So what do we think pupillometry is good for? I would love to hear your thoughts. For me, pupillometry is a good tool to index complex phenomena without disturbing them. Because measuring cognition and perception almost invariably implies interrupting or altering them. And perhaps mind wandering is a good example, because the moment I ask you whether you're mind wandering, you're obviously no longer mind wandering. So I think pupillometry may be a good objective tool to get insight into this delicate cognitive or perceptual phenomenon. So what's driving pupil size changes? The way I see it, the pupil is basically governed by two kinds of signals. And one is brightness, which is not light, and I'll explain why I prefer this formulation. And the other may be summarized by referring to locus coeruleus activity, because this is probably a good way of summarizing a plethora of cognitive, emotional, physiological phenomena that have activity in this small subcortical nucleus as a common denominator. And as a vision scientist, I've been mainly interested in brightness-related responses, but I've recently had a chance to study some of the pupil modulations of this second kind. And my motivation there has primarily been related to work in animal models. So it seems that the literature on animal models is getting more and more interested in pupillometry, so that this tool has the potential to become a common tool, a common language between us studying humans and people working with mice and other models. For example, this is a study where pupil size was measured in mice simultaneously with invasive recording of cortical activity. And among the several important findings that they have, there are two interesting observations for me. One is that pupil diameter undergoes spontaneous oscillations over time. And second, that when these oscillations are in the dilation phase, that's accompanied quite systematically by increases in the excitability of the cortical neurons, which may be a result of the norepinephrine release through activation of the locus coeruleus. Now, in humans, we do know that pupil diameter fluctuates spontaneously too. For example, in a resting state kind of situation where there are no changes in the stimulus or the task, the pupil will undergo very slow, spontaneous fluctuations. The movie here is much faster than it should be. And typically, the temporal frequencies of these oscillations are below 1 Hertz. That's sometimes called the pupillary hiatus. So one interesting question to me is, can we link these oscillations to variations in cortical processing, a little like people working with mice are doing? And we do not have the analysis ready for the experiments that are trying to address this question directly, but I thought I could provide two examples of studies that give indirect indications that yes, spontaneous pupil oscillations are informative of a person's cognitive or perceptual state, and so they are not just physiological noise. So the first example is one where we measured resting state pupillometry in a very special group of participants, which are experts in mindfulness meditation. The literature is rich with suggestions that this type of meditation affects brain activity and even brain connectivity. But that's usually estimated by comparing brain activity before and after months, weeks of meditation practice. However, measuring what happens during the act of meditating is very hard because, like I mentioned before, it is the kind of task that is inevitably disturbed if you start asking questions to the participant, but also, I mean, imagine placing a meditating subject into an MR scan or with an EEG cap, all those kind of situations that put people in an awkward position, in an uncomfortable sometimes position, are bound to be interfering with the meditation act. Pupillometry, on the other hand, really is minimally invasive. And for this particular study, we used a particularly lightweight pupillometer, which is called Pupil Labs, which basically consists of a pair of thin glasses and a portable laptop. I have to say resolution is not great in this system, but the advantage is that it allows for recording in virtually any positioning, so sitting, standing, even laying down. And also, it can be done in any setting, including an important Buddhist meditation center, which is not far from our university and is actually collaborating with our university. So we were able to recruit 23 expert meditators trained in mindfulness meditation. And we studied them over a short period of 1 hour, during which we acquired pupil recordings in samples of about 2 minutes. And we recorded an initial baseline recording, after which we invited participants to perform a short mindfulness meditation session with their eyes open, during which we recorded again samples of pupils' recordings. And the end of the soundtrack signaled to the participants that it was time to end the meditation session, and we followed this up with a short final post-meditation recording. In addition to the meditators, we recruited 20 participants with similar age and gender distribution, but no meditation training. And they completed the same protocol, except they were asked to just relax and listen to the soundtrack during session. We analyzed pupil traces using Fourier analysis, which basically means we decomposed the temporal variations of pupil size in terms of periodic oscillations of a range of frequencies. And we only checked frequencies up to about 5 Hertz, which is about the fastest oscillations that pupils can physiologically undergo, where faster changes are usually recording artifacts. The results show that there is a clear increase in the amplitude, especially of the slowest pupil fluctuations during meditation, which is this red trace, compared to the baseline recordings before and after, which is this gray trace. The bar plot quantified the power in this low frequency range, the hippus range, and shows this increase during the act of meditating that was specific for the meditators. It was not observed in the controls who were just relaxing during the soundtrack. And it was also specific for this hippus range, because if we looked at the slightly higher temporal frequencies in the delta range, we found that there was no systematic difference between the meditation session and the pre and post baselines. We also checked data quality indicators like fixation stability and number of blinks, and found that these were strictly matched across conditions and across groups. The only other variable that differed between the during and the before-after sessions was pupil diameter, but this was similar in the meditators and the controls, and it probably reflected an unspecific effect of light-dark adaptation. And remember, this was done in outside the lab, so luminance could not be precisely controlled, and participants would not spend half an hour dark adapting before the experiment. So we find that there is a specific effect on the slow, low-frequency fluctuations in the hippus range that is associated with the act of meditating. It is modulated during meditation. The interpretation of this effect is, of course, very open. Perhaps it's relevant to mention that in a previous study, we used a very similar resting state pupillometry paradigm, and we found that hippus frequencies, amplitude of hippus, were specifically and reliably associated with the sensory plasticity phenomenon, which suggests a link between these slow pupil oscillations and cortical excitability or cortical plasticity. Which may be an interesting way of looking at the effects of meditation on brain activity. However, it is obvious that we need more studies to understand the origins and substrates of these pupil rhythms, pupil oscillations, and to evaluate their association with a range of cognitive tasks. For here, we're basically comparing a very complex status, such as mindfulness meditation, with a relatively unspecific control, which is the instruction to relax. Which gives me the opportunity to move on to the next study, because when we say relax, when we are asked to relax, we actually engage in a different cognitive task, which is called mind-wandering. Mind-wandering has been studied in association with pupillometry, but unfortunately different papers have given opposite results. So we tried our hand at this problem using a paradigm that has been extensively studied by a colleague of mine at our sister University of Florence, which is summarized here. So the structure of the task is more complex because it involves multiple time scales. The basic structure is provided by a vigilance task, which is a very monotonous sequence of screen refreshes every 2 seconds. Screens have horizontal bars. Very infrequently, the bars are vertical, and the only task that the participant have is to press a button when this easily discriminable stimulus, vertical bars, is presented. And this happens on very few trials, less than 5%. So that's the first structure. Every 2 seconds, the bars change, and this will generate a small pupil constriction in response to the transient. Now there is a second structure on top of this, which is that every half a minute or so, together with the bars, there will be a cue phrase, couple of words, emotional, positive or negative, or neutral, with an emotional content, or neutral, to which participants had to give no explicit response, but of course, because they will increase the average luminance of the display, they are expected to generate a pupil constriction, a light response. And then finally, there is a third structure, which is that every couple of minutes or so, the test was interrupted and participants were questioned by the experimenter, asking whether they were focusing on the task or they were thinking of something else, mind-wandering. And if so, could they relate the contents of their thoughts to any of the keywords they had seen during the experiment? Previous research by Manila Vannucci and others had shown that these cue phrases are conducive to mind-wandering episodes, facilitate the occurrence of these episodes. And we used our pupil recordings to basically ask two questions. If there is a pupil change signature that distinguishes mind-wandering reports from on-task reports, and distinguishing cue phrases that were reported to be triggering mind-wandering from the other cue phrases. So to address number one, we ignored the basic trial structure of the vigilant task, and we aligned pupil recordings to any of the interactions and then looked back several seconds from this interaction. We found that for interruptions that were reported to be associated with mind wandering, so people reported that in the preceding few seconds, they were not focusing on the task, but were thinking of something else. There was a progressive pupil dilation, which was more pronounced than that observed when people were stopped and they reported being on task. So that's the red versus the green lines. In addition, if we looked at the cue phrases and separated out those that were reported to be triggering mind wandering in red, and those that were not reported to be triggering mind wandering in green, we found that there was a progressive pupil dilation after cue phrases that had triggered a mind wandering episode. So overall, we find that mind wandering is associated with a progressive pupil dilation unfolding over several seconds of time. This was observed in a set of 50 observers, and we basically replicated these findings in a second experiment, very similar, with some 30 people, which was had the same vigilance task structure, the same cue phrases. The only difference is that we did not have any preset interruption, but simply asked participants to monitor themselves and stop the task at any time that they caught themselves not focusing on the task anymore. They caught themselves mind wandering. Participants, very surprisingly to me, were honestly stopping themselves to report that they were not doing the task well. They were not focusing on the vigilance task. And at which point, when they stopped themselves, they were questioned by the experimenter with the same questions that we asked in the first study. So we asked them whether they were, since they were not focusing on the task, were they distracted by some external noise or disturbance, or were they mind wandering? And if they were mind wandering, were they thought related to any of the cue phrases they had seen during the experiment? We basically did the same analysis as before. So let's start with the analysis of the cue phrases. Again, we aligned pupil recordings to the presentation of cue phrases that were either reported to be triggering mind wandering in red, or they were other control cue phrases. And we actually checked that the emotional content of the cue phrases could be matched between the two. And again, like in the previous study, we found that there was a progressive pupil dilation over several seconds following a cue phrase that triggered a mind-wandering event. And similarly, we also found a progressive pupil dilation that led up to a self-caught interruption, where people reported being mind-wandering. And this was very pronounced compared to the relatively stable pupil recordings that we saw at control time points. So basically, these are the time points after a self-interruption when people had just restarted the task, and so they were very likely to be focusing on the task. So to sum up, we found that mind wandering is reliably associated with a pupil dilation. Like I mentioned, this is not always the case in the literature. And my take on this discrepancy is that a key difference may be the baseline task that we are comparing mind wandering with. So in our case, we find pupil dilation during mind wandering episodes. And this could be because mind wandering is associated with a higher cognitive or emotional load compared to the monotonous vigilance task that we use. However, imagine if the baseline task was monitoring some busy airport traffic. Well, then slipping into mind wandering could entail a reduction of load, and this could lead to a consequent constriction of the pupil. Perhaps this is a bit simplistic, but I think it makes the point that context is expected to strongly affect pupil recordings and the direction of the effects. But does this reduce the potential usefulness of pupillometry? I do not think so, because we've seen that within a given context, this vigilant task, for example, pupillometry is reliably associated, pupil size changes, in this case dilation, is reliably associated with mind wandering episodes. And we've seen it in 2 different experiments and some 80 people. To wrap up this part, it is exciting to share this line of studies on very slow pupil size changes unfolding over several seconds, which we found to be informative of cognitive processing and the mental contents of our participants. Our findings are preliminary in the sense that we know relatively little of the mechanisms generating the pupil modulations that we find. They are also preliminary in the sense that I think there is a great applicative potential to be explored, but we haven't even started exploring this, basically because we lack the competencies for doing it. For example, in the mind wandering experiment, we've done relatively simple analysis where, knowing the kind of cognitive processes that the participants reported being engaged with, we've looked at differences in pupil diameter. Exploiting the applicative potential, though, would require reversing the logic and looking at the pupil diameter over time and being able to decode what people are thinking, what people are doing based on the variations of pupil diameter, which might be possible, but we haven't actually done that yet. And it would be fun to hear if in the audience there is anybody who has ideas on whether this would be possible and how would which would be a sensible way of looking at this kind of questions. Okay, so in the last bit of my talk, I would like to shift gears a bit and move the focus back to my sort of comfort zone, which is perception and attention. And I'd like to present an example study where we used another non-trial based pupillometry, which is based on continuous psychophysics approach. And the study that I'd like to show was developed in collaboration with clinically oriented colleagues who are interested in perceptual differences across individuals, and particularly in relation to autistic traits. For perception in autism is notoriously different, but also notoriously difficult to characterize. For example, there is reported reduced susceptibility to illusions in autism, but that's according to some, not according to all. There is a tendency to focus locally as opposed to a global focus, but this, according to some, is related to the task and the context. We've noticed that research on this topic has often, though not always, used relatively complex stimuli like letters and faces. And we thought that if we had the chance to give a contribution in this line of studies, it would be by using much simpler stimuli that are part of the basic vision science toolset. And so we selected moving dots. This is the kind of stimulus we concentrated our attention on, which is made of two clouds of dots, white and black, moving in opposite directions. What's interesting about this stimulus is that if you look at it for some time, you may get the illusion of a cylinder rotating in 3D. So basically a transparent cylinder. And the illusion is bistable, where you may see it rotating clockwise with the white dots in front or counterclockwise with the black dots in front. So as a participant, you may have this oscillation perception. We thought this could be a good stimulus to expose inter-individual differences, in particular differences related to autistic traits, because we went in with a clear prediction. If you are a person with low illusion susceptibility and a tendency to focus locally, then maybe you will fix your attention on individual dots or individual clouds of dots, and you will fail to see the rotating cylinder at all. On the other hand, seeing the illusory cylinder and its bistable direction of rotation, we thought it requires distributing attention globally to all the dots, front and rear. And we predicted this would be much easier in people with less autistic traits. So we measured 50 people with varying degrees of autistic traits. And this we measured with a tool which is called the AQ score, the Autistic Quotient Score, which is a simple questionnaire that gives you a score. The score goes between 0 and 50. Above 30 or so, you typically see people with a diagnosis of an autistic spectrum disorder. In our sample, we did not have any clinically diagnosed patient, but we did see a fairly wide distribution of scores. So as a participant, you would be asked to use the keyboard to track the direction of rotation of the cylinder, and that's what we recorded here with the bistable oscillations. And meanwhile, we also recorded pupil diameter. Now, to our surprise, we found that all the persons that we tested were all very similar in that they all saw the illusory cylinder, they all experienced the bistable perception, and even if there were differences in the mean duration of the perceptual phases, so in the speed of the bistability, these were completely unrelated with the autistic traits, with the AQ scores. However, most interestingly, we found that pupillometry results did show a pattern that was consistent with our predictions. So let me show you how we analyzed them. We took the pupil recordings, the continuous pupil recordings, and we chopped them up into epochs, which were aligned to the perceptual transitions. So when people changed from seeing clockwise to counterclockwise and vice versa. And we recorded these into judgments of white dots in front or black dots in front by simply recording the responses. These are the average traces across all our participants, and we clearly see a pupil modulation that depends on the perceptual phases that participants were experiencing. And this is consistent with pupil diameter being modulated based on the luminance of the front surface. Now remember that the physical luminance, retinal illumination here was always the same, always constant. So the fact that there is a pupil modulation indicates that pupil size is being regulated based on perception or attention, which is why I said at the beginning that pupil diameter is regulated based on a brightness signal, which is perceived luminance. And we actually have, we and many others have very good evidence that this is indeed the case, that luminance is not the only driver of pupil light responses, but perception and attention also have an important contribution. So that's a basic finding. There is a modulation in pupil size. And like I said, this is the average across participants. What's interesting is that we found that this pupil modulation, consistent with attending to the front surface, was really only seen in a subset of our participants. So we took this pupil modulation that I've highlighted here with blue and quantified it for each of our 50 participants, and we plotted this value against the AQ score of that participant. And we found that there was a clear difference in pupil traces depending on the perceptual phase, only in individuals with relatively high autistic traits, with relatively high AQ scores. However, in participants with low autistic traits, low AQ scores, there was no such modulation. The pupil was always the same, quite irrespectively of whether the white or the black dots were perceived as on the front as forming the front surface. And this is in line with the concept that participants with higher autistic traits tend to have a more local focus on the front surface, whereas participants with low autistic scores tend to have a more global focus, comprehending both the front and the rear dots surface, and therefore fail to modulate their pupil diameter based on the perceptual report. So once again, what we find is a dissociation between perceptual reports and which were not correlated with the autistic scores and the pupil diameter. And really what this suggests is that it is not the ability to see the cylinder in depth or the ability to see the bistable illusion that is correlating with the AQ scores. It's only something that is contributed by the pupil diameter variations, which may be indicative of the way attention is distributed over the stimulus. And this is confirmed by a follow-up experiment where we completely got rid of the illusory and bistable aspects of the stimulus, and we disambiguated the motion of the dots by using disparity, retinal disparity. So with a mirror stereoscope, which is a system of mirrors, we presented the dots so that they were at different depth planes, and therefore disambiguated the motion, right, clockwise and counterclockwise. This way, all the participants were reporting the same kind of perceptual oscillations. However, we still found a pupil modulation, and we still found a correlation between the size of the pupil modulation and the AQ scores. So this is an example where we have a dissociation between the information we gather from pupil recordings and the information we gather from subjective perceptual reports. And this added to a fairly long list of studies where we find a similar conclusion, where we find a dissociation between pupillometry and psychophysics. I won't have time to go into these specific studies, but I'll mention that it is often the case that our pupillometry measurements give results that are in line with the predictions of perceptual variability with autistic traits. And that's for illusion susceptibility, but also for the use of prior information like priming effects. But this is also true outside the field of autistic traits, it is, for example, true in a paradigm where we looked at binocular rivalry and the effects of attention. We presented it this year at VSS, Vision Science Society, where we found that binocular rivalry, again, another bistable phenomenon, could be tracked by looking at modulations of pupil size. However, perceptual reports, perceptual oscillations, and pupil oscillations showed a systematic, qualitatively different effect of attention, meaning that perceptual reports were affected by attention, but pupil recordings were not affected by attention. And we think we should look more into these dissociations. We're very interested in this, and we think that pupillometry can give an important contribution as a complementary information compared to the perceptual reports to gather more insight into perceptual inter-individual differences in perception, but also basic visual perceptual phenomena like bistable perception and its modulations with attention. But so to conclude, I think I've shown a few examples of pupillometry studies where we took a continuous pupil recording approach and we tried to extract components of the variance related to the perceptual or cognitive tasks under study. Our analyses are not very sophisticated, and yet we were able to get a lot out of the pupil size. Not only does pupil size provide information in situations where direct questioning would be difficult, like the Mindwandering case, but it actually provides information that is qualitatively different from that gathered through questionings or psychophysics, and in some cases, more meaningful. To us, to me, this is all very exciting, but perhaps it is important to mention an enormous difficulty with working with pupillometry. And that's the fact that pupil size is a 1D variable. It's a sluggish 1D variable, which means that this single number that we record over time is actually the integration of a very large number of influences. And disentangling these influences is a complex phenomenon, complex problem. And I think making progress on this requires precisely the kind of joint effort that this workshop is all about. So I would like to thank very much the organizers for getting us together and all of you attendees. Of course, I'd like to thank the funding bodies, and particularly thanks my colleague and the people in the lab who made the research possible. Thank you.